I want you to imagine um, trying to learn the deepest, most complex secrets of modern biology. But, not from some dense, heavy textbook that puts you to sleep.
Right, definitely not a textbook.
Yeah. Imagine learning the fundamental rules of life from a viral YouTube pop song parody.
Which, I mean, sounds completely absurd on the surface.
It really does. But that is exactly our mission for this deep dive today. We are going to explore a really fascinating piece of media. It's a song called Evo-Devo by AcapellaScience.
And we have a meticulously detailed line-by-line breakdown of the lyrics to go through.
Exactly. Because this song isn't just a catchy tune. It's actually a masterclass in this concept called consilient thinking.
Consilience, yeah, that's such a great word.
It is, right. It's
Yeah.
that incredible aha moment you get when you combine multiple previously totally separate scientific fields into one unified theory.
And in this case, we're talking about evolutionary developmental biology, or, Evo-Devo for short.
Evo-Devo.
Right.
So to set the stage for why this is such a massive deal, we have to look back at the history of biology. Because for a long time, we had this really good understanding of evolution, right? Like Darwinian change over time.
Absolutely. He knew the what's of evolution: the fossil record, natural selection.
And on the other hand, we had embryology, development. We understood how a baby grows from a single cell.
Right. Scientists could watch a fertilized egg divide under a microscope and grow into a frog or a fish.
But combining those two things, that was the holy grail, because that combination finally answered the ultimate biological question: how do completely new body shapes actually evolve?
Yeah, it's the bridge. For decades, the traditional framework, what we call the modern synthesis, it was like having all the raw materials for a building.
Okay, yeah, like the bricks and the mortar.
Right. And development, the embryology side, that was the construction crew. But for the longest time, nobody knew how those two communicated.
It's like having the materials and the workers, but nobody has the architect's master blueprint.
Exactly. Evo-Devo is the discovery of that blueprint. It connects the materials to the workers.
Evo-Devo brings them together to ask this foundational question. Basically, how does evolution actually tweak the building process to create entirely new forms of life?
Because you can't just look at the final adult animal to understand evolution, right? Like, I think of it as reverse engineering software.
Oh, that's a great analogy.
Yeah, if you want to know how a piece of software evolved from version 1 to version 10, you don't just stare at the user interface.
No, you have to open up the terminal.
Right. You have to look at the underlying source code and figure out how the original hackers tweaked specific lines to change what the program actually does.
And the song actually starts by introducing us to those original biological hackers.
The very first lyrics, it's literally a roll call.
Yep.
It totally mimics a hip-hop track where the artist shouts out the producers and the legends, to set the tone.
It does. It immediately signals that this is rooted in serious science.
Yeah.
These are the absolute titans of the field.
Let's break those names down real quick for the listener. So we start with Huxley.
Right, which is pointing to the deep roots of evolutionary theory. It's likely a dual nod, actually.
Oh, to both Huxleys.
Yeah, to Thomas H. Huxley, who was a 19th century biologist, famous for being Darwin's bulldog.
Nice nickname.
Right, and his grandson, Julian Huxley, who was super instrumental in the mid-20th century in forging the modern synthesis of evolution.
Which was bringing Darwin's natural selection together with genetics.
Exactly.
And then we get Gould. I mean, Stephen Jay Gould is just unavoidable if you study evolution.
Completely unavoidable.
He really challenged the idea that evolution is just this slow, smooth, uniform march of progress. He forced everyone to view it as a deeply historical, messy process.
Messy, constrained, with these sudden bursts of rapid change.
Right, and then the more modern architects follow right after that.
Yeah, so Carroll refers to Sean B. Carroll, who is just an absolute giant in modern Evo-Devo.
He wrote Endless Forms Most Beautiful, right?
Yes. That book basically introduced this entire field to the general public.
Yeah.
And then Davidson is Eric H. Davidson, a researcher who spent, honestly, his entire life painstakingly mapping out the incredibly complex gene regulatory networks of sea urchins.
Sea urchins, of course.
Hey, they're useful. He basically drew the very first highly detailed wiring diagrams of the biological computer.
And all these names, these legends, they were all obsessed with the exact same fundamental mystery, which the song drops in the very next line.
See one cell divide and decide on 1,000 fates. Did you ever figure how they know?
The 1,000 fates problem. This is the core of it all for me. Every single human being, every whale, every fruit fly, it all starts as one single fertilized egg, a zygote.
Just one cell.
And it divides into 2, then 4, then 8. But eventually they have to stop just cloning themselves and start specializing. They have to differentiate into 1,000 different fates.
Exactly. Some become neurons firing in your visual cortex.
Right, and some become acid-secreting cells in your stomach or the tough keratinized cells of your fingernails.
It's mind-boggling when you really think about it.
But the sticking point for me has always been the identical manual.
The identical DNA.
Yeah. Every single one of those cells contains the exact same sequence of DNA, the identical blueprint. So if the manual is the same, how does the cell reading it know which chapter it's supposed to execute? How does one decide to be an eyeball and another decide to be a toe?
Well, the information has to come from outside the DNA itself. It comes from the environment of the embryo.
Okay, how so?
The song explains his core concept beautifully right away. We are built of modules, combined in a planned-out way. Each new piece must be told where to go.
So, positional information.
Positional information, exactly.
Yeah.
A cell doesn't just passively sit there. It is constantly listening to its surroundings, trying to figure out its spatial coordinates.
But let's get into the mechanics of that. How does a microscopic cell listen to spatial coordinates? I mean, it doesn't have a tiny biological GPS antenna, right?
Well, actually, in a way, it kind of does. So as the early embryo develops, specific clusters of cells start acting as signaling centers. They secrete these specialized chemical molecules called morphogen, and these morphogens diffuse outward through the surrounding tissue, which creates a concentration gradient.
Ah, okay, a gradient.
Yeah, so the cells closest to the signaling center, they're bathed in a very high concentration of this chemical.
They're drenched in it.
Exactly. The cells a little further away receive a medium dose, and the cells on the far edges receive almost none.
Okay, I follow that.
And the cells themselves have receptor proteins on their outer surface that physically bind to these morphogen molecules. By basically measuring the proportion of the receptors that are currently activated, the cell calculates its distance from the source.
So it's kind of like sitting in a massive stadium, right? And the speaker system is blasting a high-pitched tone from the main stage.
Oh, that's a good way to visualize it.
If the sound is deafening, you know you're in the front row. If it's a medium volume, you're somewhere in the middle sections. And if it's just a faint hum, you know you're up in the nosebleeds.
Exactly.
So your DNA is the rule book that everyone has in their pocket, but the volume of the sound tells you exactly which page to open to.
That's precisely it. If a cell senses a high morphogen concentration, it triggers internal pathways that say, okay, I am located at the very front of the animal, activate the head building module.
And if it senses a low concentration?
It says, I am at the back, activate the tail building module. And the key word there is module.
Right, because the song says built of modules combined.
Evolution doesn't build a body as one giant, continuous, unbroken block of clay. It builds modular segments that can be repeated or tweaked or repurposed.
Like your spinal column.
Perfect example. It's just a series of repeated vertebral modules. And this modularity means the overall instruction set doesn't need to be infinitely complex.
Which transitions perfectly into how we conceptualize that instruction set in the first place. The song uses this incredible metaphor next. Phenotype the interface for mouse and man. Genotype the files and the subprograms. What then are the switches, circuit boards, and boot code?
We have officially moved away from the old architectural blueprint analogy and straight into computer science.
Yes, hardware and software.
And the terminology maps so perfectly. The genotype is the underlying sequence of your DNA. So in the computing metaphor, your genotype is the raw machine code. It's the hidden files, the deep subroutines just sitting quietly on the hard drive.
And the phenotype?
The phenotype is the physical manifestation of that code. It's your eye color, your height, the physical shape of a mouse's ear. The phenotype is the graphical user interface. It's the screen output that the world actually interacts with.
So when I tap the colorful app icon on my phone screen, I'm interacting with the phenotype.
Right.
But the thousands of lines of unreadable text executing behind the scenes to make the app open, that's the genotype.
Exactly. But, a hard drive sitting on a desk doesn't do anything on its own.
Right, it's just stored data.
It requires an operating system to execute it, which is why the song asks about the switches, circuit boards, and boot code.
And the chorus immediately answers that question. Every gene directed by a signal key code, proteins that can activate, enhance, or veto.
This is where we dive into the logic gates of life.
Let's get into it.
Biologists call these gene regulatory networks, or GRNs. For a really long time, the focus of genetics was almost entirely on the structural genes.
The ones that build things.
Yeah, the sequences that actually code for physical building blocks like collagen or muscle fibers or hemoglobin. But Evo-Devo revealed that a massive, critical portion of our DNA doesn't build structures at all.
What does it do?
Its only job is to manage the other genes.
It's the middle management of the genome.
It literally is. And these regulatory genes produce specialized proteins called transcription factors. Their entire function in life is to float around inside the nucleus, find very specific sequences of DNA, and physically bind to them.
Just latch right on?
Yep. And when they bind, they act as a switch. They can activate a gene, calling over the cellular machinery to start reading it, or they can enhance it, basically turning up the volume so the gene produces a massive amount of its product, or they can veto it.
Vetoing meaning they act as a silencer, right?
Exactly.
Let's look at the mechanics of a veto for a second. Are they just like blocking the machinery from getting to the DNA?
Physically, yes. The transcription factor will lock onto a regulatory region of the DNA right next to the target gene, and its physical bulk prevents the RNA polymerase, which is the enzyme that reads the gene from attaching.
It just gets in the way.
It clamps down and silences the gene entirely. You can't read what you can't reach.
I love the electrical grid analogy for this. So the structural genes are the actual physical light bulbs, the neon signs, the heavy-duty stadium floodlights.
Right.
But the transcription factors, the regulatory network, those are the relay stations, the dimmer switches, the timers, and the emergency shutoff valves.
That's a great way to put it.
Because you absolutely need those switches to ensure that the massive stadium floodlights don't accidentally turn on inside a tiny residential bedroom at 3 in the morning.
And building on that electrical grid idea, these switches don't operate in a vacuum. A single timer doesn't just control one bulb.
Right, the song describes them as calculating in a network labyrinthal where the heart and liver and the hands and feet go.
It's a labyrinth because it's a cascading hierarchy of logic gates. So gene A might produce a transcription factor that turns on genes B, C, and D, but then gene B produces a protein that turns on 5 more genes. And meanwhile, gene C produces a protein that circles back and actually vetoes gene A to shut down the original signal.
Wow, so it turns itself off.
Yes. It's a massive cascading calculation happening in real time inside the cell. It takes the input, those morphogen GPS coordinates we discussed earlier, crunches the numbers through these labyrinthal logic gates, and outputs a physical structure, like a liver or a foot.
But, dealing with software architecture brings up a frankly terrifying reality. When a modern operating system has a bug, it crashes, your screen goes black, you reboot the computer, and you go on with your day.
Minor inconvenience.
Right. But biological software isn't modern.
Not at all.
The song points this out explicitly. With circuits so deeply built upon, they're older than the Paleozoic era, baby.
We are talking about deep time here.
Yeah, how old is the Paleozoic?
The Paleozoic era started over 500 million years ago.
500 million years.
Yeah. One of the most staggering revelations of Evo-Devo is that the core biological software governing how a brain splits into hemispheres or how an appendage forms wasn't written recently.
Right.
It wasn't invented when mammals first evolved. It wasn't even invented when the first fish swam. This code was established in the very earliest, weirdest, multicellular animals in the primordial oceans.
And it just stuck around.
It has been passed down, largely unchanged, for 500 million years.
It's the ultimate legacy code.
Exactly.
And anyone who has worked in software engineering knows you do not touch the deep legacy code.
Never touch the legacy code.
Right. It's the foundation of the entire architecture. If you mess with a subroutine written 50 years ago that the entire banking system relies on, everything collapses.
And in biology, that collapse is catastrophic. Which brings us to a really devastating real-world case study hidden in the next lyric.
Oh, this part is so fascinating. The lyric is, in a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes, as this cyclopic sheep knows.
The cyclopic sheep of Idaho.
Just a crazy story.
It's from the 1950s, and it perfectly illustrates what happens when ancient legacy code experiences a fatal crash.
Set the scene for us.
So, sheep farmers in the high alpine meadows of Idaho started experiencing this horrific wave of birth defects in their flocks. Pregnant ewes were giving birth to lambs with severe facial deformities. In the extreme cases, the lambs were born with a single, perfectly formed eye right in the center of their forehead.
They were literal cyclops.
Literally.
It sounds like pure mythology. And initially, I imagine everyone assumed this was a genetic curse, right? Like a terrible random mutation sweeping through the flock's DNA.
That was definitely the leading hypothesis at the time. But the U.S. Department of Agriculture sent researchers out there, and after nearly a decade of painstaking work, they realized it wasn't genetic at all.
It was environmental.
Exactly. They noticed the timing of the pregnancies coincided with the sheep grazing on a specific high-altitude plant called Veratrum californicum.
Commonly known as the corn lily, right?
Right, the corn lily.
But how does a pregnant sheep eating a weed cause the embryo to grow a single eye? What is the actual mechanism happening there?
Well, the corn lily produces a potent steroidal alkaloid, which is basically a chemical defense mechanism against being eaten.
Makes sense.
The scientists isolated this toxin and named it cyclopamine.
Because of what it did to the lambs.
Right. And what cyclopamine actually does is it molecularly jams one of those ancient Paleozoic gene regulatory networks we've been talking about.
Which one?
Specifically, a network called the hedgehog signaling pathway.
I know biologists have a reputation for quirky naming conventions, but naming a foundational piece of evolutionary code after a small, spiky mammal feels like a bit of a stretch.
Yeah, the name actually comes from fruit fly research back in the 1970s.
Okay, how did that happen?
Scientists Christian Nusslein-Volhard and Eric Wiesias were intentionally mutating fly embryos to see what would happen, basically trying to map out the developmental genes.
Right.
They found one mutation that caused the fly larva to be covered in this dense coat of spiky structures, making it look like a tiny hedgehog. So they naturally named the gene hedgehog.
Okay, that's fair.
And when researchers later found the equivalent homologous genes in vertebrates, like mice and humans, they just kept the joke going. They named them things like Indian hedgehog, desert hedgehog.
And the most famous one, Sonic Hedgehog.
Sonic Hedgehog.
Literally named after the Sega video game character.
Yep.
But despite the funny name, this pathway is a matter of life and death. How does Sonic Hedgehog actually work in a developing embryo?
Sonic Hedgehog is one of those crucial morphogens we talked about earlier. During the very early stages of embryonic development, there's a structure running down the exact midline of the embryo called the notochord.
The notochord?
And it begins secreting Sonic Hedgehog protein, which creates that chemical gradient spreading outward.
Like the stadium speakers?
Exactly. This gradient acts as the GPS coordinate for the center of the face. It tells the surrounding tissue, here is the exact midline. The brain tissue needs to divide into a left and right hemisphere around this line, and the optical tissue needs to migrate to either side to form two separate eyes.
So the cyclopamine toxin from the corn lily enters the pregnant sheep's bloodstream.
It crosses the placenta.
And it physically interacts with this pathway.
It's a chemical blockade. Cyclopamine physically binds to a specific receptor protein on the surface of the embryo cells, a receptor called smoothened.
Smoothened?
Right, and by locking onto smoothened, the toxin permanently jams the receptor. The cells are suddenly deaf. They cannot hear the Sonic Hedgehog signal anymore.
The GPS system goes completely offline.
Completely. And without that high concentration signal screaming, this is the midline, divide here, the developing forebrain never splits into two hemispheres. It just develops as one single continuous lobe condition called holoprosencephaly. And because the brain doesn't split, the optical field doesn't divide. The eye tissue just coalesces into one large central eye in the middle of the face.
It's a tragic, really haunting image, but biologically, it is a staggering revelation.
It really is.
Because the lamb wasn't missing the structural genes to build a cornea or a retina or an optic nerve. The bricks to build the house were all sitting right there.
Perfectly intact.
But the regulatory switch, the ancient master command that tells the body where to put those bricks, was chemically muted.
Which proves the song's point. Where they go, calamity goes. You don't get a healthy, novel, one-eyed species of sheep. You get a fatal system crash.
The rules of development are incredibly rigid.
Extremely. The legacy code is deeply conserved because it absolutely has to be.
Okay, so we've seen the catastrophic calamity of blocking a master signaling pathway. But that naturally begs the reverse question. If a single chemical signal can dictate the placement of an eye, what happens if scientists artificially trigger that signal?
Ah, yes.
What if you take the master switch and forcefully flip it on in a part of the body where it absolutely does not belong?
This takes us right into one of the most mind-bending experiments in the history of biology.
The song sets the stage perfectly. See, down they cascade like a domino. Like you and I, Drosophila, the path that makes us optical was laid a long, long time ago.
Drosophila melanogaster, the humble fruit fly.
The undisputed workhorse of genetics.
Oh, absolutely. They breed rapidly, they're cheap to maintain, and their genome is incredibly easy to map and manipulate. But the crucial lyric here is the next one. The path that makes us optical was laid a long, long time ago, back before we blew up the Cambrian like a bomb.
The Cambrian explosion, we're going back roughly 540 million years.
Right. If you look at the fossil record before the Cambrian period, life is mostly simple, soft-bodied, and relatively uniform.
Sponges, worm-like things.
Yeah. Then, in an evolutionary blink of an eye during the Cambrian, there is this massive explosion of diversity. Suddenly we see complex body plans, hard shells, jointed appendages, and crucially,
The sudden appearance of complex eyes.
exactly.
And for decades, the assumption was that because a human eye, which works like a single camera lens, focusing light onto a retina, looks and operates so fundamentally differently from a fly's eye,
Which is a multifaceted compound honeycomb of thousands of tiny lenses.
right, because they are so different, people assumed evolution must have invented the concept of vision completely independently over and over again.
It was called convergent evolution. The structures are so completely alien to one another that they couldn't possibly share a common origin.
The blueprint for a camera is not the blueprint for a honeycomb.
Right. But Evo-Devo introduced a concept called deep homology, which blew that assumption entirely out of the water.
How so?
When geneticists started hunting for the regulatory switches that control eye development, they found a gene called PAX6.
Tell me about PX6.
Well, in humans, if you have a mutation in the PAX6 gene, you were born with severe defects in your iris and retina.
Okay.
When they looked at mice, a mutation in the mouse version of PAX6 resulted in mice being born completely without eyes at all.
Wow.
And meanwhile, over in the fruit fly labs, researchers had long known about a gene they simply called eyeless.
Because flies with a mutated version had no eyes.
Exactly.
And when they finally had the technology to sequence the actual base pairs of DNA,
They realized that the fly's eyeless gene and the mammalian PAX6 gene were essentially the exact same sequence of DNA.
that's crazy.
They were homologs. Evolution did not invent the eye from scratch dozens of times. Deep in the Precambrian era, before the Cambrian explosion even happened, evolution invented the master regulatory switch for build an eye here.
And that single switch was passed down, conserved through 500 million years, to virtually every animal that can see.
Every single one.
Which sets up arguably the funniest yet most profound lyric in the entire song. Now my eye protein can make you see out of your bum-bum.
It sounds like a crude joke, I know.
It really does.
But it is a direct reference to the Walter Gehring experiment from 1995.
Let's talk about Walter Gehring.
Gehring and his team in Switzerland wanted to test the ultimate power of this deep homology. So they took the PAX6 gene, specifically the version of the gene found in a mouse.
A mouse gene.
Yes. And they used genetic engineering to splice it into the genome of a fruit fly.
Okay.
But they attached it to a special promoter that allowed them to turn it on wherever they wanted.
So they essentially took the mouse's master switch and wired it into the fly's biological computer.
Exactly. And they artificially flipped that switch on in the fly's imaginal discs.
Which are what?
They are the clusters of cells in the larva that are destined to become the fly's legs, its wings, and its abdomen.
So they commanded the fly's knee to build an eye.
And it worked.
It actually worked.
Yes. As the fly metamorphosed, fully formed, structurally complex eyes began growing out of its legs. Eyes sprouted on its wings. Eyes grew on its abdomen.
That is just, it's horrifying but amazing.
And these weren't just useless blobs of pigment either. They had fully developed photoreceptors and corneal lenses. They even hooked up electrodes to these leg eyes and found they were physiologically responding to light.
The imagery is just like a surrealist nightmare.
Very much so.
But there is a massive conceptual trap here that I think we need to navigate carefully for you listening. The team used a mouse gene.
Right.
They injected the master switch from a mammal. So did the fly grow a tiny, squishy, camera lens mouse eye on its leg? Or did it grow a multifaceted, honeycomb fly eye?
And that is the defining question of Evo-Devo. The fly grew a fly eye.
But why? If the command came from a mouse, why doesn't it build mouse hardware?
Because PX6 does not contain the structural blueprints for a mammalian retina or a lens. PX6 is a high-level executive command. It is a transcription factor whose only job is to sit at the very top of the hierarchy and say, initiate the eye-building subroutine.
Ah, okay.
When the mouse PX6 protein was produced in the fly's leg, it successfully bound to the fly's DNA because the switch mechanism is identical across species.
The lock and key are the same.
Exactly. It flipped the master switch. The local fly cells received the executive order and they said, understood. We are building an eye.
Yeah.
But when they went to their hard drive to execute the subroutine, the only structural files they had were the genes for a fly eye.
So they grabbed the fly bricks, fly mortar, and fly glass to follow the mouse's order.
Yes.
It's like walking into a traditional Italian restaurant, grabbing a megaphone, and shouting the French word cuisiner. The chefs understand the universal command to start making food, but they are going to use the tomatoes, garlic, and pasta they have in their own kitchen. They aren't going to suddenly bake a French baguette just because you yelled in French.
That's a perfect analogy.
Thank you.
And this completely revolutionizes how we understand the mechanics of evolutionary change. If a single regulatory switch can trigger the cascading development of an entire complex organ, you don't need to wait for a million random mutations to slowly assemble an eye from scratch. You just have to tinker with where and when the master switches are deployed.
Which is the core thesis of the song, laid out right in the next verse. Mostly don't evolve in the genes of the genome. Safer the mutation aimed at regulation. Keep the building blocks and swap their activation.
The song is essentially arguing that evolution is lazy, but in a highly efficient way.
An evolutionary shortcut.
Exactly. For a very long time, the dominant thought was that physical changes in animals occurred because the structural genes themselves mutated. Like the gene that makes a protein mutates, making a slightly different protein, which changes the shape of a bone.
But Evo-Devo says.
Evo-Devo says actually mutating the actual structural building blocks is incredibly dangerous.
Because of pleiotropy, right?
Right, pleiotropy. The idea that one gene affects multiple traits.
Let's break that down.
Okay, so the structural proteins in your body are heavily reused. If you have a random mutation in the structural gene that codes for collagen, you might slightly change the shape of your jawbone, which could theoretically be beneficial. But that same mutated collagen is also used in your heart valves, your skin, your blood vessels, and your tendons.
Oh, I see.
By changing the brick itself, you compromise the structural integrity of the entire organism. It's almost always a lethal mutation.
So safer the mutation aimed at regulation. If you can't change the brick, you change the architectural blueprints.
You tinker with the regulatory DNA, specifically regions called enhancers. A single structural gene might have 5 different enhancer switches attached to it.
Okay.
One switch turns the gene on in the jaw, one turns it on in the leg, one in the heart. If a mutation randomly breaks the enhancer switch for the jaw shape changes.
But the heart is fine.
Exactly. The gene still functions perfectly normally in the heart and the leg because those specific switches are untouched.
You are keeping the building blocks but swapping their activation. It's totally like Lego.
How so?
Well, evolution isn't waiting millions of years for a random chance to invent a newly shaped, perfectly molded plastic brick. It's just rewriting the instruction manual on how to assemble the standard bricks we already have.
That is a great summary.
But, we've seen this Lego manual rewriting go wrong in the lab with the fly eyes. The song brings up another famous natural mutation, though. Homologs evolve from repeats in the schema. Switch a couple bases in the proper places. You'll be watching flies grow legs out of their faces.
Legs out of their face. This is referring to the Antennapedia mutant, which is another really famous fruit fly discovery.
Legs instead of antenna.
Exactly. And to understand it, we have to define what the song means by schema and homologs. It's talking about Hox genes.
Hox genes, let's unpack those. They are kind of the true architects of the body plan, right?
Yes, they are. Hox genes are a specific family of master regulatory genes that lay out the head-to-tail axis of an animal.
Okay.
They tell the embryo, this segment is the head, this segment is the thorax, this segment is the abdomen. And what's incredible about Hox genes is collinearity.
Collinearity, what does that mean in this context?
It means they literally sit on the DNA chromosome in the exact same physical order as the body segments they control.
Wait, really?
Yeah. The gene for the head is first, the gene for the thorax is next, and so on down the line. And they all share a specific 180 base pair DNA sequence called a homeobox.
Which does what?
It allows the protein to physically lock onto the DNA helix.
Okay, so with that setup, what exactly goes wrong in the Antennapedia mutant?
It's a regulatory typo. A mutation happens in the enhancer region of the Hox gene that normally controls the development of the fly's middle leg segment.
Okay.
This tiny typo causes that leg-building Hox gene to accidentally activate in the segment of the head that is supposed to grow antenna.
Oh, no. And because it's a master switch, it just executes the subroutine blindly.
Blindly. The cells in the forehead receive the executive command to build a leg. So instead of delicate sensory antenna, the fly develops 2 fully formed, jointed legs jutting right out of his forehead.
Just sticking out of its face.
Yeah, it is bizarre to look at. But mechanically, it proves the rule. The genes to build the leg are perfectly healthy. The structural bricks are fine. They're just deployed to the wrong ZIP code because of a tiny mutation in the regulatory switch.
It's just so mechanical. And this mechanical understanding is why the song claims we need an update to our fundamental theories of biology. The lyrics say, "Stick around for modern synthesis, the sequel. Only by combining can a new theory grow evolution and development, amigos."
Right.
So why did the modern synthesis need a sequel?
Because it treated the development of the animal as a complete black box. The modern synthesis, which was finalized in the 1930s and 40s, was a monumental achievement.
No doubt.
It combined Darwin's natural selection with Mendel's genetics. It mathematically proved that traits are inherited through genes, and natural selection acts on genetic variation within populations.
I mean, that sounds pretty comprehensive. What was missing?
The actual organism.
Ah.
Yeah, the modern synthesis looked at the beginning, the genes in an egg, and it looked at the end, an adult animal interacting with its environment. But it completely ignored the middle.
The messy part.
The messy, miraculous process of how the egg actually becomes the adult. It essentially said DNA goes in, an adult animal comes out, and we don't really need to know how the sausage is made.
To be fair, they probably didn't have the tools to know.
Right, exactly. They didn't have the molecular tools to see inside the black box back then.
So Evo-Devo is the sequel. It pries open the black box. It says you cannot fully understand how an animal's form evolves unless you understand the physical constraints and possibilities of how it is built as an embryo.
Evolution and development, amigos. They are completely intertwined.
You can't understand the software's evolution without understanding the compiler.
That's a perfect way to phrase it.
Okay, so we've spent a lot of time in the laboratory talking about fruit flies and genetic engineering. Let's take this Lego manual rewriting concept out into the real world. Let's look at how animals actually weaponize this code to survive and adapt in the wild.
Let's do it.
The song transitions to reptiles here. Look at how our spinal segments generate a neat row built on a molecular clock. One cycle, one vertebra. Speeding up its rate is snake's developmental cheat code.
The molecular clock of the spine. This is a process called somitogenesis.
Somitogenesis.
Mhm.
Okay.
As any vertebrate embryo grows, whether it's a mouse, a human, or a snake, it elongates from head to tail.
Ah.
And as the tissue grows backward, there is a literal rhythmic chemical clock ticking inside the cells.
A clock made of chemicals. How does a biological clock actually tick? Like, what is physically happening?
It relies on a negative feedback loop, often involving a signaling pathway called Notch.
Notch.
So a gene turns on and starts pumping out a specific protein. As the concentration of that protein rises, it eventually reaches a threshold where it circles back and binds to its own gene, shutting it off.
So it kills its own production.
Right. And then the protein naturally degrades over time.
Mhm.
Once it's gone, the gene is unblocked, it turns back on, and the cycle starts again.
It's an oscillating circuit.
Exactly. This creates a pulsing wave of gene expression traveling through the embryo tissue. Tick, tick, tick.
That's brilliant. And how does that build a spine?
Every time that chemical wave peaks and crashes, it triggers the cells in that zone to pinch off into a distinct block of tissue called a somite. Yeah, one tick of the clock equals one somite. And those somites eventually harden into the individual vertebrae of the spine.
Okay, so for a human, that clock ticks at a very specific rate relative to how fast the embryo is growing. It ticks enough times to carve out, 33 individual vertebrae before the tail-end growth phase stops?
Right.
But a snake can easily have over 300 vertebrae. How does it manage that? I'm assuming it didn't painstakingly evolve 260 brand new structural genes for bone.
Not at all. It uses a phenomenon called heterochrony, which literally means different time.
Different time.
It's a change in the timing of developmental events. Snakes evolved a regulatory mutation that simply sped up the oscillation of that Notch molecular clock.
So it just ticks faster.
The wave pulses much faster. Tick, 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 churning out tiny block after tiny block at a rapid-fire pace while the embryo is elongating.
They just turned up the speed dial on the conveyor belt?
That's the developmental cheat code.
Hmm.
They didn't invent new bones. They just altered the frequency of an ancient oscillating network.
It's so elegant. But, evolution isn't always about gaining things. Sometimes it's about losing them.
Very true.
The song follows up immediately with, "That and where a lizard's feet grow, they turn off distal aminos."
This refers to creatures like skinks or glass lizards. Yeah, if you look at a glass lizard, you would immediately call it a snake. It has no legs.
Right.
But structurally, genetically, and phylogenetically, it is a lizard that just happens to be legless.
Mm.
Evolutionary biologists have noted that the loss of limbs happens incredibly frequently across different lineages, and it happens very fast in evolutionary time.
But why is it so easy to lose a limb? I mean, building a leg seems incredibly complicated. Shouldn't dismantling it be just as hard?
Well, dismantling the structure would be hard.
What do you mean?
Slowly mutating and deleting the millions of base pairs of DNA that code for bone, muscle, and cartilage. That would take forever. But Evo-Devo gives evolution an out.
The regulatory switches.
Exactly. To build a leg, you need a cascade of regulatory switches. Switch A turns on switch B, which turns on the build toes subroutine.
And the song specifically mentions distal aminos. We should clarify that for you listening. Distal means situated away from the center of the body, right? So your shoulder is proximal, your fingertips are distal.
Right.
And aminos refers to amino acids, the building blocks of the regulatory proteins.
Exactly. So if a lizard population moves into an environment where having legs is a massive disadvantage, say they start burrowing through tight, packed sand where legs just cause friction.
You need to get rid of them fast.
Right. So evolution just needs a single point mutation in one of the enhancer switches controlling the distal leg development. It breaks the switch. It deploys a veto. Structural genes to build the toes and the feet are still sitting perfectly intact on the hard drive, but the transcription factor required to press start is broken.
The distal aminos are turned off, that
Yeah.
is. It's just incredible to realize how modular and logically tweakable life really is. It sort of takes the mystery out of it, but replaces it with an even deeper appreciation for the actual mechanics.
I completely agree.
But there is a lingering question here about how we actually know all of this. Like, we're sitting here talking about invisible chemical waves and microscopic transcription factors binding to DNA and proteins vetoing other proteins.
It all sounds very theoretical until you see it.
Exactly. How does a scientist actually see this happening? This takes us to the lab techniques the song highlights near the end. Map out a gene with a glow tag. Kill it with a morpholino, short for oligomorpholino baby.
The song provides a really cool peek into the molecular toolkit that made Evo-Devo possible. Because you can't just look at a fruit fly embryo with a magnifying glass and see a gene turn on.
Right.
You need glow tags.
So what exactly is a glow tag doing?
The most famous example is GFP, or green fluorescent protein. This was a protein originally discovered in a species of glowing jellyfish.
Jellyfish. Of course.
Yeah, and what's magical about GFP is that it is an autocatalytic barrel shape. It folds itself into a little lantern that glows bright green under UV light, and it doesn't require any other chemical enzymes to work.
So how do you use a jellyfish lantern to map a fruit fly's genome?
Genetic engineering. Scientists take the specific regulatory enhancer switch they want to study, say the switch that turns on the eyeless gene we talked about,
Okay.
and they splice the GFP gene directly downstream of it in the DNA. They create a transgenic embryo.
Oh, I see where this is going.
As the transparent embryo develops, the exact millisecond that the regulatory network flips that switch, it doesn't just produce the eye protein, it produces the jellyfish protein.
Oh, wow. So you can literally sit there looking through a fluorescent microscope and a tiny cluster of cells in the leg or the head will suddenly light up in neon green.
Exactly. You are physically watching the spatial pattern of the code executing in real time.
You are watching the invisible network become visible.
It's how we map the labyrinth.
That is beautiful. And what about the next tool? Kill it with a morpholino. It sounds so aggressive. And the song clarifies it's short for oligomorpholino.
Yeah, a morpholino is a specific type of synthetic molecule called an oligonucleotide.
Oligonucleotide.
Right, an oligo is just a short sequence of nucleic acids, like a tiny custom-built snippet of synthetic DNA or RNA. Morpholinos are designed to test the function of a gene by temporarily knocking it out.
But how does a synthetic snippet of DNA kill a gene's expression?
It's all about steric hindrance,
Yeah.
physical blocking.
Like the veto we talked about.
Similar,
Yeah.
Let's say you see a gene glow green and you hypothesize, I think that specific gene is the command to build a beating heart.
Okay.
To prove it, you manufacture a morpholino that is perfectly complementary to the messenger RNA of that target gene. You inject it into a single-cell zebrafish embryo.
Zebrafish, another classic lab animal.
Yep. When the embryo tries to read that gene and sends the mRNA out to be translated into a protein by the ribosome, the morpholino acts like molecular glue. It binds tightly to the start sequence of the mRNA.
It jams the zipper.
Exactly. The massive ribosome machinery comes along, hits the morpholino roadblock, and completely stalls out.
Huh?
The protein is never built.
Wow.
You have effectively killed that line of code without permanently mutating the DNA, and then you just watch the embryo grow.
And if the zebrafish develops perfectly normally but simply lacks a beating heart?
You have absolute proof that you found the heart switch.
It's reverse engineering through strategic sabotage. You turn off the breaker switches one by one until you figure out which one controls the kitchen lights.
That's exactly what it is.
It's brilliant. And this brings us to the grand finale of the song, which zooms all the way back out to the macro level. The lyrics say, "Voyage of the beagle body plans evolve when proteins steer their genome. In this manner, life's beauty glows aesthetica in vivo."
Aesthetica in vivo, beauty in a living thing.
It's a gorgeous summary.
It's a beautiful way to end the journey. It is the ultimate summary of consilience. We started with Charles Darwin on the voyage of the beagle in the 1830s. He looked at the macroscopic world, the subtle variations in the beaks of Galapagos finches, the different shapes of tortoise shells.
Right. He observed the output, the phenotype.
Yes, and he deduced the selective pressures shaping them. But he had absolutely no mechanism.
He didn't know what a gene was, let alone a morphogen gradient.
Right, and now, almost 2 centuries later, Evo-Devo has taken us all the way down to the atomic level. We know exactly how body plans evolve.
It happens when microscopic transcription factor proteins bind to regulatory enhancers to steer their genome.
It happens through the shifting of ancient, deep homologous circuits that were laid down before the Cambrian explosion.
You know, there's sometimes a fear that when science digs this deeply into the mechanics of life, when we reduce the majesty of biology to logic gates, repressor proteins, and chemical clocks, that it somehow strips away the magic.
I hear that a lot.
Yeah, that it makes life feel cold and robotic. But honestly, walking through this song, it does the exact opposite for me. Understanding the labyrinthal circuits, knowing that a human eye and a fly eye are linked by a shared molecular master switch across 500 million years of deep time.
It's profound.
It really is. It makes a single fruit fly sitting on my kitchen counter feel like a living, breathing miracle of engineering. It makes the magic tangible.
I completely agree. The complexity is what makes it beautiful. The diversity of life on Earth isn't just a chaotic soup of random, disconnected accidents.
Yeah.
It is a unified, highly logical, ongoing symphony played on an ancient genetic instrument.
Okay.
The deeper we look into the code, the more elegant the programming appears.
It's been an incredible journey unpacking this today. We started with a YouTube parody song and ended up deconstructing the fundamental source code of embryology and evolution.
We covered a lot of ground.
We really did. We learned how a single cell relies on morphogen gradients to find its GPS coordinates. We saw the catastrophic system failure of cyclopic sheep when environmental toxins jam the legacy code of the hedgehog pathway.
And we marveled at the deep homology of the PIX6 master switch, growing a fly eye on the leg.
Right. And how evolution prefers the safety of tinkering with regulatory switches over breaking structural building blocks. We explored the molecular clocks churning out SNC spines and the lab tools like GFP and morpholinos that let scientists watch and manipulate the code in real time.
It is a massive amount of biology hidden in a 3-minute pop parody. And that profound realization leads me to a final thought I want to leave with you, the listener.
Oh, this is the big question.
Yeah, think about this. We've established that the biological operating system running inside you right now is ancient, blind, and totally reliant on random, slow mutations.
The slow path of natural evolution.
But, as we just discussed with the glow tags and the morpholinos, we aren't just observing it anymore. We possess the literal tools to tag, read, and intentionally switch off these master genes at will.
We found the control panel.
Exactly. So, if natural evolution is just the blind tweaking of these switches, what does the next phase of life look like now that the organism has finally learned how to hack its own boot code?
It's a completely new frontier.
It really is. Thank you for joining us on this deep dive into the consilient, labyrinthine, and deeply beautiful world of Evo-Devo. We'll catch you next time.